Hello, anatomy and physiology students. This is Mrs. Johnson again, and we are going to continue our notes on the physiology of a muscle. These notes begin on 53 of your packet of notes. And if you remember our first video, we talked about how a muscle was stimulated by the brain, how the muscle actually received a message from the brain, and how that acts potential travel down the neuron, causes acetylcholine to be released by the neuron, and how that acetylcholine traveled across the synaptic cleft to the muscle fiber, and the muscle fiber took that acetylcholine and allowed sodium to rush inside the muscle cell, and that huge influx of positive ions, sodium ions, causes that action potential to continue through the muscle fiber and continue the message. If you want, you could watch this video and review that first part of the muscle physiology. But today we're going to create a muscle contraction, create movement. We're going to be looking at this video to really help us understand how that movement is created. Here's a few more animations that may help you understand how a muscle contracts. This process of a muscle actually creating a movement, the muscle contracting, is called a sliding filament theory. And the first thing that happens is once the muscle fiber receives a message from the nervous system, the first thing that happens is calcium ions are released. Can you remember where our muscles store calcium ions? That's right, that's sarcoplasmic reticulum. If you can remember, the sarcoplasmic reticulum is a specialized smooth ER that our muscle fibers have that solely stores calcium just for this process to create movement. Here's this picture you guys looked at in way back in the beginning of the anatomy of a muscle, and you can see all the myofibrils, the actin and myosin inside the muscle tissue, and if you look right there you can see the sarcoplasmic reticulum is just laced in between, and that's what releases the calcium. Now let's talk about what that calcium actually does. How does that calcium actually help create a movement? I'm going to move to this animation because I think this animation does a much better job of explaining what happens in the sliding filament theory. What we're looking at here is a sarcomere. A sarcomere starts at a Z disc and ends at a Z disc, and it has thin filaments and thick filaments. And if you remember, a thin filament is an actin, and the thick filament, this green here, is a myosin. Okay? And so what the first thing that happens is after a muscle receives the nerve impulse, calcium is released. Here comes the calcium. See that green ball? Okay, and that calcium binds to tropomyosin, and what it does is, it binds to troponin. And what happens is when that calcium binds to troponin, it causes tropomyosin to move out of the way. Did you see that movement? It's very small, but there was something that changed. So when that calcium, and I'm backing it up a little bit, the calcium binds to the troponin and creates tropomyosin to move out of the way. What that's doing is it's allowing this myosin to be able to bind to actin. It's unblocking actin. So when calcium is present, troponin and tropomyosin move out of the way, and the next thing that happens is that myosin head is able to make a cross bridge, is able to bind with actin. Okay, this is called a cross bridge where myosin binds with actin. Do you see there's another one over here? Okay, and what's going to happen next with ATP present here, ADP, and there's a phosphate there, with those present, it will have the energy to create a movement. Did you see that contraction? Did you see how the myosin was able to pull the actin and create a movement? Now of course here it goes again. ATP causes that myosin to bind to actin and create a movement, all with the presence of calcium and allowing that troponin and tropomyosin to be out of the way. Now of course this process where myosin grabs actin and pulls, grabs actin and pulls, grabs actin and pulls, this process can happen in 1,000th of a second. We are breaking it down very, very slowly, but this process happens thousands of times in 1 second. And this is over and over and over again, all the myofibrils in a muscle causing this sliding filament theory to occur creates a muscle movement. I like this picture because it basically kind of breaks down the whole process. If you look up at the top left corner, you see the axon communicating with the muscle. This gets the rest of the picture. How the acetylcholine is released. It's going across that synaptic cleft. And then a whole bunch of sodium is rushed in. If you read that, it says sodium is rushed in, and it causes the sarcoplasmic reticulum to release the calcium. The calcium goes to the myofibrils, the actin and myosin, and binds to troponin and causes tropomyosin to get out of the way, which then allows this myofibril, this red thing down here, to bind to actin, creating a cross bridge. And then that myosin and actin slide against each other. The myofibril pulls the actin and creates a contraction. Now, a few things happen to that sarcomere after the sliding filament theory. Again, if you look at this top picture, you see the sarcomere, you see the Z disc, you see the actin, you see the myosin. This is when a muscle is relaxed. You have an H zone. The H zone again is where there is no actin, thin filament. Okay. After a contraction, that's the bottom picture, you can see that the whole sarcomere has shortened. And you can imagine if all the sarcomeres and all of your muscle fibers and all of your muscles shortened that much, it would create a movement. It would cause the whole muscle to contract. Another thing that's changed is that the actin, the H zone, has now disappeared. You can see that the actin molecules are actually overlapping each other in the middle, and that causes that H zone to completely disappear. Now, do you guys remember back in Unit 2 when we looked at skeletal and cardiac muscles and we noticed that it had striations, stripes? Well, those stripes are caused by the presence of these myofibrils, these A band. Okay, the thick myosin causes those striations, causes those stripes. Check out this animation. It will also help you understand what happens to a sarcomere. In this animation, you can actually see two sarcomeres. 1, 2. Again, there's the Z line, the Z disc, and each one has myofibrils and myosin and actin. And after they walk along each other and contract, there, the myosins are pulling the actin closer and closer and closer into the H zone. Do you see how the H zone is disappearing? And the whole sarcomere got shorter. This is how a muscle contracts.